Good afternoon, all, and welcome to our webinar, Rapid Detection and Removal of Biofilm Made Easy Using New Technology, held in partnership with our friends and gold business partner, AFCO, a ZEP company. My name is Tom Wojno, Senior Vice President of Innovation and Member Advancement at IDFA, and I want to thank you all for joining us today. The presence of biofilm in food processing plants has been one of the biggest challenges that food processors face on a regular basis. It can lead to food product contamination and shorten a product shelf life. Our program today will discuss the importance of adopting an effective biofilm detection procedure and implement an effective sanitation program aimed at hopefully eliminating it from all our food processing equipment and environmental surfaces. Before we begin, I have just a few housekeeping items to cover. All lines are muted. We encourage you to submit your questions via the Q&A feature at the bottom of your Zoom dashboard at any time during this presentation, and we will do our best to answer them at the end at our Q&A segment. Our presentation is being recorded and will be housed on our website in the Knowledge Center under the IDFA Virtual Programming tab. If you experience any difficulties or technical issues, please email our membership at idfa.org email address, and we'll do our best to help you. With that, it is my pleasure to introduce our speaker today, Adel Makdesi, a senior corporate microbiologist with AFCO ZEP. I want to thank you for being with us today. Thank you for sharing your expertise. And with that, I will turn the presentation over to you.
Thank you, Tom, for the introduction. And I want to thank the International Dairy Foods Association for this opportunity and all the attendees for their time. And hopefully they will learn some stuff from this presentation that would help them in the real life. So today, as Tom said, we're going to talk about the rapid ways to detect and remove biofilm using new technologies. But before we talk about that, I would like to talk a little bit about biofilm. So what's a biofilm? Biofilm is a natural habitat. It's made by microorganisms, including pathogens like Listeria and Salmonella and E. coli. So the biofilm itself consists of course, a huge number of microorganisms. Not just microorganisms, but there is also proteins and fats and sugar and carbohydrates and other organic matters that go into the matrix or the build of or the structure of the biofilm itself. Biofilm usually can be formed by microorganisms within 8 to 24 hours. So if the microorganisms is in an optimum condition for forming a biofilm, it will start forming it, and within 8 hours to 24 hours, you'll have a biofilm on its surface. And that's, as you know, I'm sure we all heard about the biofilm, how dangerous it is to have in a food processing plant because it contaminates food products and shortens the shelf life, as Tom said. Biofilm forms rapidly, mainly in wet environments. So whenever the environment is wet, like floors and pipes, transfer pipes and water systems, biofilm forms really quick in there. And we need to get rid of it. So as you see in this slide here, we've got examples of a biofilm. I'm not sure if you guys seen it before or not. I'm sure you've seen it. By looking at these pictures, you're probably shaking your head and saying, yep, I've seen that. And that's what biofilm is. So you see it on a wall here in a form of like mold and yeast and bacteria is all together forming this black slimy stain. Also in the drains, it could be brownish, could be yellowish, it could be black as it is here in the picture. In the dairy and all the drinks processing plants, you can find biofilm also in the transfer pipes for CIP systems. You see that yellow slimy film inside the pipe, the second picture on the right, that's a biofilm inside a transfer pipe. Also, we can find them in like a cooking facility where they have hoods on the top and all the moisture goes into the hood and sucks it up. And it can also, these hoods, if not cleaned and sanitized on regular basis, they can form a biofilm, and the biofilm will start, the humidity will condense on this biofilm and start dripping down on the food that is being cooked underneath. So it's really very dangerous to have biofilm. So how is the biofilm form in an environment, whether it's a food processing plant or outside on the streets or in your mouth? So biofilm can be found anywhere. Like I said, where there is food and humidity, or constant food and humidity, biofilm forms really rapidly. Let's say in a food processing plant, when a bacteria like Salmonella or E. coli or Listeria enters a facility, for example, via a raw material. Raw materials are considered, contaminated. That's why we call it raw material. And remember, biofilm can be formed by any microorganisms or by any bacteria. So when, for example, we have a steak or milk or whatever, we put it on a surface like a belt or a table, all right? So some of the bacteria that are on the steak or in the milk will stick to the surface. Once it sticks to the surface and it starts feeling that there is something missing in there, and this bacteria doesn't want to die. So what they start doing is producing what we call the AHLS. So we have here an example of an E. coli cell producing this complex chemical. I'm not going to say the name of it, but we call that AHL. This AHL is being produced by the bacteria when the bacteria is ready to form a biofilm. What this AHL does is sends a signal to the surrounding bacteria. There are bacteria around that particular bacteria that are producing the AHL. So it's a chemical that produced in the environment, in the air, and other bacteria can sense it. So it tells the other bacteria that we are here around you. We are neighbors and we need to do something to rescue our life. So this will detect other bacteria around, and it tells these bacteria that we are starving and it is time to form a biofilm before we die. So this AHL, it triggers certain genes that are activated within these bacteria to start forming a biofilm. So when these bacteria, all the neighboring bacteria, get ready to produce a biofilm, it will produce it. How is the biofilm produced? When bacteria starts multiplying, they produce a gluish material, like you see under the microscope around this bacteria here in the bottom, around the black bacteria. This is what we call ESP, or we call it extracellular polymetric substances. That's produced by the bacteria, and these substances have some benefits for the bacteria, okay? So this substance is part of the matrix that protects the bacteria or make the bacteria stick to the surface that they are forming the biofilm on, okay? So the more they produce babies, let's call them baby bacteria, the more of these ESPs are being produced around the bacteria and above the bacteria to protect themselves. So they use it to attach themselves to the surface and bind their cells together. And if you look at the top picture, you'll see the bacteria are next to each other. They are binding together with this polysaccharide ESP. Well, it's actually a very chemical resistant, this ESP or extracellular polymetric substances, which consist mainly of polysaccharide or complex sugar. Okay, so that complex sugar is very chemical resistant and difficult to remove from the surface. And that's why these bacteria start using it to protect themselves, coat themselves with it, stick themselves to the surface, right? And so they can protect themselves from any environmental conditions and chemicals that are going to come later when they clean. So once these bacteria start multiplying, that will multiply layer after layer. So the surface will be filled with bacteria. And then it comes another layer on top of them of baby bacteria and another layer. They keep multiplying and forming the biofilm. So the biofilm becomes thicker and thicker with time. So if you don't remove the biofilm from the surface when it's very thin, it will become really difficult to remove with chemicals. We're going to talk about ways to eliminate the formation of biofilm. That's why biofilm in the can varies in the thickness between microns and inches. So microns is invisible. When there is a biofilm sometimes on a surface, it's invisible. When it is very thin, it's the beginning of the formation of the biofilm. So that's why it's very difficult to detect or to tell if the surface has a biofilm or not, okay? Sometimes that biofilm gets really thicker and thicker and thicker, like a water holding tanks that are not being cleaned for years and years. If you touch it on the inside, it will be slimy. You empty the tank from the water, you can see the slime, and it gets really thick. So you can tell there was a biofilm in there. While the biofilm is multiplying and to different layers, microorganisms start capturing some organic matters, including proteins, carbohydrates, minerals, calcium, all kinds of things that they need for their survival, and capturing it and bring it into the structure of the biofilm. Remember, it's a layer of polysaccharide that has all kinds of things in it now, from carbohydrates to proteins to minerals to bacteria, millions of bacteria feeding on all this food that they bring in from the next day's processing. So after you clean, you process again, you put food on that surface that has a biofilm, and you cannot see it. They get some of that food, get it into that matrix of the biofilm, and they store it in there. So as you see in the picture, the black and white picture in the bottom here, so there is a structure to the biofilm. And there is tunnels in there. It's called organized tunnels, okay? So these organized tunnels is what allows the bacteria to move freely in these tunnels to bring food, and also for, to inhale and exhale and use oxygen or not, whatever that they need, it goes through it. So if you look at this biofilm here, is under the microscope, you will see all type of bacteria in there. Some of them are small, some of them are a little bigger. And sometimes the same biofilm will contain yeast. And you can tell the yeast from the bacteria by the size. The yeast size is 7 times bigger than the bacteria, as you can see. So that big cell is a yeast in there. But again, this biofilm has organized tunnels or channels that are important for the survival of this biofilm. And the longer you leave it on the surface, the thicker the biofilm gets and more difficult to remove, and the more bacteria will be in it. So in a biofilm itself, once the biofilm is formed, there are the bacteria in there, or microorganisms. That are two types of bacteria that go into the biofilm. The first type is what we call the planktonic microorganisms. These are the bacteria that float on the surface of the biofilm. Okay, so you can see it from this outside. When you put a biofilm on under the microscope, what you see is the planktonic microorganisms that are on the surface. And these can be easily killed because some of them floats in a solution. Let's say we have a biofilm in a pipe, and the pipe has water in it, or a water tank. And so these planktonic microorganisms are any of those bacteria that are on the surface of the biofilm, and they can float in the solution and transfer to another place, go to another place and contaminate it. And that's why it's easier to kill those. The other type of microorganisms in the biofilm, we call it the sessile microorganisms. These are the microbes that are embedded and anchored into the surface by the matrix of the biofilm, which is the polysaccharide, that thing that it produces and makes it really stick to the surface and protect the bacteria. So those are embedded inside the biofilm. So if you have a biofilm on a surface and you put a sanitizer on top of it, right, you can kill some of the planktonic microorganisms that are on the surface. However, this sanitizer is not going to penetrate the biofilm and kill those microorganisms that are embedded inside the biofilm. And that's why when you clean that way without scrubbing and removing the biofilm, it's like you didn't do anything, because you still have millions of bacteria inside the biofilm that is stuck to that surface, and you cannot see it because it's very, very thin layer, like microns. It's like you have a fridge, you buy a new fridge, it comes with this plastic film that you can't see sometimes, and you leave it there for years, you don't know it exists. Something like that, much thinner than that plastic, but that's how it looks on the surface when you don't know what it is. So again, biofilm may contain large numbers, like millions of pathogenic bacteria, including Salmonella and E. coli. And this biofilm can sometimes break due to some physical agitation or a knife or a brush or something that hit the biofilm, and that biofilm will crack. Once it's cracked, it will start releasing millions of bacteria into that surface and contaminate the products when you put food products on it when you're processing. So that's why it is very dangerous to have it in a food processing plant. So also biofilm can move, believe it or not, it can move especially in a fluid system, like a CIP system or tanks or pipes. It can move by what we call on the top, like streaming. Pieces of that biofilm will detach itself and start streaming, goes with the flow of the solution, or detaching, or seeding. Some biofilm produce like get cracked from the force of the water inside the pipe. It can crack and start some of the bacteria start going into the solution or into the milk or into whatever you are processing and contaminate that milk. We call that seeding dispersal. Also, we have rolling. Pieces of that biofilm will start rolling. It's a slime, like a ball. It's keep rolling, rolling, rolling. And when they move, they contaminate other parts of the CIP system, or when it goes, the solution goes onto the floor, it will contaminate also the floors, okay? And another way is called drippling, like you see in here. So there are different ways in which biofilm can move and contaminate other areas in your food processing plants. Once you have it in one place, it can contaminate other areas. Now, the challenges or the problems that we have mainly with biofilm is visibility. When the biofilm is really thin, like microns thin, it will be very difficult to see. So when you test a surface and you knew that you have a biofilm, from the microbial count, you do a microbial count, standard plate count, or selective agar or petri dishes, whatever, and you notice that you have millions of bacteria or too numerous to count on your petri dish, and you never had that before. You know that you have a biofilm, and you look at that surface, you go back on that surface, you look at it, it looks clean to the naked eye, yet you have a biofilm on it. So this is a visibility problem with the biofilm. Why is it important? Because, of course, if you have a biofilm, you want to make sure to remove it. So the next day when you process, the biofilm wouldn't stay there and contaminate the food, right? So this is a challenge for food processors, even in pipes, the same way. Now, if you decide to remove the biofilm, which you have to, once you have a biofilm, you need to remove it. It's very labor intensive. Remember what I said about the matrix, that gluish polysaccharide material they produce to stick themselves to the surface with it? This is very chemical reaction, very chemical resistant. And when you apply detergents and sanitizers, it's not going to remove it. Traditional cleaners and sanitizer are not going to remove the biofilm. So what you need to do is scrub that surface. If you have an external surface, you have to scrub every inch of that surface to guarantee that you remove the entire biofilm. Sometimes you miss some of the inches, sometimes you miss some part of that surface, and you have still biofilm on it, and you're still in trouble. So this is another challenge, which is the labor that takes for removing a complete removal of a biofilm on a surface, okay? So also in a CIP system, if you have it in a CIP system, you have to run chemicals in the CIP system, and and agitate that CIP system for a long time, 4 to 8 hours, with some oxidizing sanitizers to remove it. But luckily, there are other procedures that we're going to talk about to rapidly remove biofilms from vertical surfaces and from CIP and COP systems. So the tip to effective sanitation program, so you don't have a biofilm in your processing plant, are proper use of detergents and manual scrubbing for complete removal of soils from the surfaces. So when we clean every day, you need to use the right concentration of the chemicals, and you have to use manual scrubbing on difficult to clean areas and also difficult to reach areas. If you leave any food in there, that means there are some bacteria left on that surface, and these bacteria will start calling again for forming a biofilm, and that's a trouble. So if you use the right chemical, you scrub, you will not have a biofilm. Pay attention to hard to reach areas. There are hard to reach areas that you say, okay, it's all inside there, I'm not going to clean it, I can't reach it. These are the dangerous areas that promote the formation of biofilm, because nobody is touching this bacteria in that area there. So bacteria will multiply, form a biofilm, and contaminate everything you have, so all your work is gone. Disassemble equipment when possible. So every equipment that is disassemblable, you have to disassemble it and break it down and clean it and put it back together, because biofilm can form if there are cracks between the different parts and you don't clean it properly. Always use recommended concentration of sanitizers. If a sanitizer, call on the label for example, 200 ppm, and the dispensing system for the sanitizer is dispensing it at 100 or 50 ppm, and you don't know because you're not making titrations for the chemicals before you start every day, which is a mistake. So what happens in this case, these bacteria on the surface that you are applying 100 ppm on them or 50 ppm instead of 200, they'll start building resistance to the sanitizer. So when you use it the next day and the next day at 100 and the next day at 100, so some of those bacteria will die, but some of them will stay alive because it's not 200 ppm. Those bacteria that stayed on the surface will start forming a biofilm because they know there's something being applied on them, which is the sanitizer at 100 ppm, that is not killing them yet, it's making them uncomfortable. So they start calling each other to form a biofilm to protect their life and continue their genes, right? So they start forming a biofilm. That's why always use the right correct concentration of the sanitizer. Flood sanitize the surfaces. Never use a pump-up sprayer. A lot of people use pump-up sprayer to just mist spray food processing equipment, the external surfaces. Don't do that. You need to flood all surfaces of the equipment with a lot of solution of the sanitizer to make sure you covered the entire surface and no bacteria are left on that surface. Oh. Okay, use acid detergents weekly to prevent the formation of protein and mineral scales. Why is that important? You do not want to leave any protein stuck to the surface. That protein can be used by the bacteria to multiply and form a biofilm. That's number one. Number two, mineral scales. Mineral scales will make the surface rough, and it has a lot of pores in it. Mineral scales have a lot of pores where these bacteria can hide in those pores and avoid the sanitizer and stay alive and start forming a biofilm. That's why when you use an acid detergent once a week, it will remove all protein buildups and all mineral scales from the surface, which prevent the bacteria from hiding and forming a biofilm. Okay, so now we talked about the biofilm. If we have a biofilm, what are the rapid ways to detect the biofilm? There are new technologies that came about to detect the biofilm on the surface. And it's very important to have those because it will give you the green light to clean or not to clean. So rapid technologies to identify areas of high risk. Of course, when you have a biofilm, you are in high risk. So we have what we call instruments or swabs. Okay, so we use sometimes the ATP, which is one of, everybody knows what ATP is, to test to see if the surface is clean or not. And why is it important to clean the surface? Again, if you leave any food, bacteria can use it to form a biofilm on that surface. So ATP can measure if there is any food or not on that surface, but the ATP has limitation because when you are testing that surface, you are choosing 2x2 area, right? On the whole piece of equipment sometimes. So you are missing the rest of the surface that you didn't swab, and you probably have organic matters or ATP on it and you don't know. Also, there are some protein testing swabs and there are some sugar testing swab. All those can test if there is any food residue on the surface, but they all swab a piece of that, like 2x2, 4 square inches of a surface that of the entire surface of the equipment. You are not going to be going and swabbing the entire pieces of equipment with those to tell if there is still organic matters or not. So these testing methods have have their limitations to be as effective as other ways. Other rapid ways to detect biofilm, we have visual indication detection technologies. There are some technologies out there that they use like a gel. Spray a gel on a surface and wait for a few minutes until the bubbles start generating. If there is any organic matters left on that surface that you cleaned and you're testing to see if you have any organic matters or biofilm in there, will start bubbling up little by little to see these tiny bubbles in that gel. And that's how you know if there is organic matters or biofilm or there. There is another technology called the SMART technology, which is a colorimetric technology which uses the persulfate technology. Okay, so this is a technology that counts on the change of the color of the gel that you spray. So there is a complete product line for this VIP verification in place product line. Some of the products are liquid and some of them are powder and others are gel. You can use any of these products. When the color is changed, we're going to talk about these technologies. When the color is changed, you can to different color after you spray it, seconds after you spray it, you can say, okay, I have microbes or I don't have, I have organic matters biofilm or I don't. So for example, there is a complete product line. Now this is, again, we have products that are chlorinated, and it's only one single component. You mix it with water, put it in CIP system, and then circulate it 10 to 15 minutes. And if the color, it changes from purple, we'll say, into a green or yellow, you know there is organic matters in there. So these first four products, these are chlorinated or non-chlorinated detergency that has an indication, color indication in it, changes the color, will tell you if the CIP line is contaminated or not. Does it have organic matter or not? Does it have a biofilm? Remember, biofilm contains organic matters. We talked about containing proteins, carbohydrates, even sometimes alcohol, all kind of things. So these are products that are being oxidized with this colorimetric product, and with change of the color tells you there is organic matter. Okay, so all products are alkaline and contain oxidizers. So it has chlorine, permanganate, and then persulfate. And oxidizing agents in there in that product when interact with the organic matters. And the color indicator, it changes the color. If there is organic matter, it will change the color. If there is no organic matters or biofilm in that CIP system, the color of that solution stays as it is. This top 4 PST product, which is, persulfate technology product, are very sensitive. They can be, they detect traces of organic matter less than 0.1 milligram per liter of organic matter. It can detect it and it will change the color of that solution. The fifth product down there is a gel. The gel color is visually, you can spray it on external surfaces, not for CIP system, but for external surfaces of tanks, belts, whatever you want to use it. And it's also very, very sensitive. It detects 1 to 2 microns per centimeter squares of glucose, if there is glucose, and also 5 micrograms per square centimeter of albumin. So these technologies are very sensitive and it's very rapid. And it's visual. You can see the surface of the solution changing color, and I will show you some examples of that. So these are the first 4 products for a CIP system. You can use any of the 4 products that we have. They have some detergency in them as well, so they can detect and clean the surface or the CIP system when you use these products. So for example, you fill the system with water and add any of these 5 products we talked about, the 4 products, not the gel one, but the liquid or powder. And you will have a solution with the color purple in it inside the system. Okay. So if the system, after 10 to 15 minutes of circulating that solution inside the CIP system, the solution stays purple, that means the system is clean and you don't have any organic matters or any biofilm or anything in there. If the color of the solution changed from purple to green, that means there is some organic matters in that CIP system and need to be cleaned again. If the color of the solution changed to yellow, that means there is a lot of organic matters, high amount of organic matters being detected, including biofilm in that CIP system, and that calls for additional cleaning step to remove all these biofilm. And we're going to talk about the cleaning. So this is for the liquid and the powder products for CIP system. Now, for the external surfaces, before we move there, we've got some pictures here of a solution that we use in a CIP system on the top left. And remember, there are some CIP systems with what we call dead ends, and they can form a lot of biofilm and organic matters on the gaskets and in the end of the dead end, because the solution is not running to remove all this through the dead end. So you have to pay attention to the dead ends and make sure they are clean. The gaskets between the pipes are all clean because they can form biofilm and slime on them. They have to be checked every 3 to 6 months and make sure that they are replaced when they are slimy and cracked. Now, in this last picture in the bottom here, we have fillers. We tested the fillers. Some fillers get contaminated or have biofilm. Some of them don't. So the bottles here, all the 4 bottles stayed clean. That means the 4 fillers in the top are clean, but the one in the middle changed to green, which means this filler is not clean enough and it has some organic matters in it. That's all. The testing happened, of course, after you clean the system, after you clean the fillers, after you clean the CIP system. You use these solutions to tell you if the system is clean or not. Okay. And then we have the 4th product that we are going to be using, which is the Smart Gel. This is a 2-part product. As you see, you'll empty this little bottle down there into that spray bottle, shake it a little bit, and spray it on a surface. Okay. So it also has the same ingredients like the other chemicals in there, but this is a gel product. We use it for external surfaces. So it's the same principle. Color integrated products or colorimetric. You spray it, it's purple. When you mix these 2 products together and you spray it, it's purple. If the color changes to green or yellow, it means there is organic matter on that surface, and I'm going to show you some examples. It gives you the results very quickly, and you can spray the entire surface of the equipment with that. We have this product comes in large amounts, 55-gallon drums, if I'm not mistaken, 20 or 5-gallon drums, pails. So you can spray the entire piece of equipment and see where it's still dirty, where that surface where it's clean. So again, the results are given within a few minutes. Like, for example, if you look at this belt here that was sprayed with this gel, where the pink is still pink, the pink when you spray the product is pink. And in the areas where it changes to little green, that means there is organic matter, and that part of the belt is not clean, and that means you have to clean it. If you look at the tank above the belt, also some areas are pink, some areas are green or yellowish, which means it has organic matters. So when it is really yellow, that means you have a lot of organic matter on that surface and you didn't clean it properly. Other areas here, we spray the gel on it. And you can tell that the surface were clean. It's all nice and pink. And some areas up in the top, it's unclean because it changed the color to greenish. So you have to scrub it again. It's a very simple and very effective way to detect the presence of organic matters and biofilm on the surfaces by using this smart technology, okay? And you can spray the entire piece of equipment with it and tell where it's clean or not clean, and it gives you a result within seconds. Okay, so now we detected biofilm in there. We have some biofilm. What are we going to do about it? We're going to clean it. We're going to scrub every inch of that CIP system. How are we going to scrub the CIP system? That's very difficult. How are we going to scrub every inch of that piece of equipment that we don't know if it's completely covered with biofilm or not? So we actually have a new technology to quickly remove the biofilm from either way, external or CIP systems. What we call that technology is called PerQuat technology. Okay, there are different types of products for that technology. It comes in a liquid form or powder form. It's a two-part product. You have to mix them at the same proportions. So if you use part A 16 ounce, you have to use part B 16 ounce per gallon of water. If you use it at 12.8 ounce, you have to put part 1, you have to put part 2 also at 12.8 with a gallon of water, and then foam it on a surface, and that will remove the biofilm. So it's approved for use in food processing plants on food contact surfaces and non-food contact surfaces. So you can use it all over the equipment, foam all the equipment with this product, cover it with foam. So you make sure that the entire surface is covered with foam, and that foam is going to remove the biofilm that you cannot see, and then rinse it with water, and it's completely now free of biofilm. How does that product work? Within 10 seconds, with 10 minutes contact time, sometimes on the equipment in the drains, you can leave it 30 minutes for more contact, right? But on surfaces like external surfaces of equipment like belts and tanks and stuff like that, you cannot leave it too long because it will dry off, okay? And when it dries off, it leaves some white residual because the product itself is an alkaline, has some alkalinity in it, and it leaves white residue. So you start rinsing it before it starts drying. So once you apply it on a surface, that foam starts breaking down the biofilm by means of hydrolysis, oxidizing, and solubilization. Okay, so there's 3 chemical functions that work together in that chemical, in that foam, to penetrate the biofilm and break it down and kill the bacteria that are embedded inside the biofilm, which we call the sessile bacteria or sessile microorganisms. Okay, so these are some graphs or different steps of how the product works. The blue balls on the top is the microsolve itself. You apply it on a surface that has a biofilm. The brownish or yellowish layer with bacteria, this is a biofilm, right? So once you apply the foam on the biofilm, the foam particles or the chemistry starts penetrating the biofilm and cracking it. That's something that alkaline detergents and caustics and stuff that you use, traditional cleaners that you use on daily basis would not do. But this product would do that kind of chemical function, which is penetrate the biofilm, and then it will break it down. If you look at the 3 pictures, it will destroy and eliminate the biofilm, breaking it down by a unique combination of hydrolysis, oxidation, and solubilization. So the biofilm is completely removed. And so you come back after that, you rinse the surface from all the foam, and the biofilm is gone. So you have a complete coverage of the surface, complete removal of biofilm without the need to scrub every inch of that surface. Unless, of course, you have a slime that you can see, and it's really thick on that surface in your plant. Then in this case, you might need to scrub to scrub the surface with brushes or green pad, and then use once a week, use this product to foam all the equipment with it to prevent any biofilm from staying on the surface, okay, before it gets that thick. So this is how the Microsolve product works. Again, it's a two-part product, and it removes biofilm quickly by contact only without the need for scrubbing. When the biofilm is still thin, you cannot see like on external surfaces of this your processing equipment. So they did a comparison of this product with per-quad technology. And they compare it to other chemicals like bleach and quat to see how they are effective in removing biofilm under the microscope. So the images on the left shows the surfaces with a biofilm under the microscope. That's before it's treated with bleach or quat and glutaraldehyde. The pictures, the images on the right are after treatment. So after treatment, you can see they still have biofilm. The top three images, you still have biofilm on these surfaces. The quat and bleach didn't do much to remove the biofilm. Now, in the bottom pictures, this is where Microsolve is being applied on the surface that has a lot of biofilm on it. And then after 10 minutes, it's being rinsed, looked under the microscope, and the biofilm is completely removed. And this is how it looks, the stainless steel kind of wavy under the microscope because you're enlarging the image so much. It looks smooth to you to the naked eye, but it's not very smooth. But there was no biofilm after applying what we call the Microsolve on that surface. So Microsolve application, it can be used on walls, drains, floors, ceilings, food processing equipment, application equipment, which is the equipment that you use to foam with, to sanitize with. They get greasy and they can be slimy, so you can foam them with Microsolve to remove that slime from the surface, which is a biofilm. Any slimy thing is a biofilm, and you have to remove it, and it contains a lot of bacteria. You can also use it in a CIP system. This Microsolve, there is a non-foaming version of this chemistry also. Any place where organic or microbial films may be present, you can use biofilm. The application can be foaming. You can foam that product. You can use it in a COP tank to soak with it parts of the equipment, pieces of equipment, and you let it sit there for 1 hour. All the biofilm is being removed. You can run it in a CIP system between 30 to 60 minutes, and it will remove all the biofilm from the pipes and the gaskets and the tanks of the CIP system. You can also use it in the drains. So the drains, you can foam them, as you see in the bottom picture on the right. You can fill the trench of the drain with the foam and let it sit there for 20 to 30 minutes and then rinse it off. And then all the biofilm and slime is gone into the drain. So if you want to use Microsolve, there are two different treatments. Okay, there is a shocking treatment. If you detect there are microorganisms or there is contamination on the surfaces, you test it for Listeria, you test it for Salmonella or E. coli on the surfaces of your equipment or on the floors or in zone 3, zone 2, zone 1, any zone, and you detect you detected microorganisms or pathogenic microorganisms in there, and you do not want to have it. So this is what when we use the shocking treatment, which is using that Microsolve at 12.8 to 16 ounce per gallon of water per part per gallon of water every day until the problem is gone. Okay, remember, biofilm could be thick. So you have to use sometimes multiple applications to be able to remove it from a surface, especially on the floors. Okay. So this, again, this treatment is a shocking treatment. You use it every day. Hopefully it won't take more than a week to remove everything. And you test after every application, you do microbial testing to see if you still have biofilm or you still have bacteria or not on that surface. And a maintenance program, which is the program that you apply to prevent biofilm from being detected on the processing equipment. After, like when you clean every day, then once a week, in the end of the week, you use Microsolve. After cleaning with an alkaline detergent, rinse it off and then foam with Microsolve and let it sit 10 minutes, rinse it off, and then you will never have a biofilm on that piece of equipment. This is a maintenance program for plants that they don't have microbial problem to keep it that way. So remember that all food contact surfaces must be rinsed with potable water before, after foaming with Microsolve. You can't leave it on the surfaces of the equipment. After you rinse it off, then you apply the sanitizer. So you use an alkaline detergent, whatever caustic or alkaline you're using, rinse it off with water, then apply microsalt because you need the caustic or the alkaline to remove the grease and the fats from the surface, okay? Because microsalt could be sometimes a little difficult to remove this thick grease on the surface. So you have to use an alkaline first, and then you use microsalt, and then you rinse it off, and then you sanitize, okay? So this is the order of how you use microsalt. And the microsalt product or the Perquat technology can be flushed into the sanitary waste system, although it contains quat and hydrogen peroxide bond together into that bridge that does all the work, okay, chemical work, which is the removal of biofilm. Because it's bond with hydrogen peroxide, it will not affect much the sanitary waste system or the wastewater plant, wastewater in the plant that you have. So it will not interfere much unless you use it every day with large amounts. Then it will start interfering with your culture that you add in the water plant system. You know, the wastewater system that you are using and that you have in the plant. So, but if you use it once a week, you will not have any problem in the wastewater system. For the floors, you can do the same exact thing. So if for the drains, if you have floor drains that are, you detected the listeria in there, you do the same thing every day, foam them with microsol until you test after that to make sure there is nothing left, and then you stop when you do not have any listeria or any E. coli or salmonella in the drain. So this is a shocking treatment. And if you want to do a maintenance treatment the same way, exactly like equipment, you just pump that foam once a week into the drain, make sure it will remove and let it sit for 20 to 30 minutes, rinse it off with water, and then you are good to go. But on a daily basis in the maintenance program, you have to use the detergent that you are using to clean the equipment. You have to use that detergent to clean your drains. You can't leave the drains dirty the whole week and then use microsalt once a week. That's not going to do it. Every day you use a detergent and sanitizer in the drain. Once a week, if any biofilm form on that drain, you use microsalt to remove it as a maintenance program. Now, again, if you detect biofilm in the drain, you have to use microsalt every day until there is no more biofilm or bacteria in the drains. This is the benefit of using foam in the drain, because you can pump the foam underground in the underground pipe and fill it up with foam, and that foam will remove the biofilm on the inside surfaces of that drain pipe. Okay, so this is really good. At 360 degrees coverage, it will remove all the biofilm from under drain pipes. If you see here, we put liquid. When you use a sanitizer solution in the drain, will cover only part of the underground pipe, not even half of it. And remember, this sanitizer, regular sanitizer, like what? It will only kill the planktonic microorganism on the surface of the biofilm. It will not penetrate the biofilm, okay? But when you use the foam, it will penetrate the biofilm at 360 degrees in the underground pipes of the drain system. Okay, so the benefits of using the Perquat technology or the microsalt: it removes the stubborn organic buildup biofilm from the biofilm from the surfaces and destroys the microorganisms that are embedded into that biofilm. The foam application will provide visual confirmation that the entire surface is being covered to ensure that the biofilm is removed from the entire surface. Extended contact time: the foam is good because it will extend the contact time. Instead of spraying just with that microsalt spraying, the solution will not foam, it will drip, will run down on the floor. So foam is good to, the foam of the biofilm is good to cover and stays there on the surface for 10 minutes to interact with the biofilm and remove it. The microsalt eliminates the need for manual scrubbing. Okay, so you just cover the entire surface with microsalt, let it sit 10 minutes, and then rinse it off. Ideal for treating difficult to reach areas. You can just shoot that foam in there and then make sure that the biofilm is eliminated in there. It eliminates any spikes in microbial count. That's when the biofilm is cracked, hit by somebody, some sanitarians, some production personnel, and then you start producing the seeding. Remember, start producing all these bacteria, and you test that surface, and it spikes with the count, microbial count. If you use microsolve, you will not have a biofilm, so you will not have these spikes of the microbial count on the surface. And extend food product shelf life, of course, when you don't have bacteria on the surface. Eliminates fruit flies in the drain. So when you apply it in the drains, all the fruit flies will be eliminated with this product, with this microsolve, which is an added benefit to using microsolve. Easy and quick. It's 20 to 30 minutes to 20 to 30 seconds or maybe 40 seconds per drain. Instead of brushing the drain, it will take 15 minutes to brush every drain and clean it, right? So this one, just pump the foam in it, 30, 40 seconds, and let it sit for 10 minutes, then rinse it off. And again, it provides a full coverage of the tank at 360 degrees around the entire underground pipe. There are different units that you can use. Any of the foaming units that you have in the plant can be used for using microsolve, right? Any of these foaming, the 5-gallon, 10-gallon, 15 gallons, and 25-gallon pump, foaming units. You can add the mixer into the drums or into the container of the foamer, foam with it, and then there are other systems that can be used. In this system we call microsolve easy blend. Okay, so there is the blue one and the green, two different chambers for the chemical. So you put chemicals in them and it will pull the chemical at the right proportion. Remember, it has to be the same amount exactly of each part goes to the water pipe, the dilutes being diluted and then applied. So there are two different systems for the microsolve that can be used, just mount it on the walls, or you can roll it around and foam with it as you go, connect it to the water hose and roll it and foam the entire plant, the walls, the floors, the equipments once a week, and you will be good to go. These are the floor attachments for the drain system, because you need to cover the drain if it's rounded. Hopefully you can pump more into that underground. You have to cover the drain. So we have a 12 inches plate. We have 7 inches plate, and there is like a foam plunger, like for small drains, to cover it and start pumping the foam into the drain through this pipe or the handle of that attachments you see there, right? So these are the different units to use, and this is a titration kit. If you want to titrate the chemical of microsolve, you can use this kit to titrate it. Now, in summary, again, detection and prevention and elimination of biofilm is made quick and easy by using this new technology. So please protect your brand and your plant by using these technologies. Detection and elimination of biofilm. All right, so now we have questions. Any questions for anybody? Tom, am I going too fast?
No, you're doing great. I'm gonna, we've got till the top of the hour. So what I'm gonna do is rattle off a few and if you wanna answer these, and then we'll try to get to the rest. If we don't get any, I'll forward them to you, Adel, and you can
answer
get to
them.
it. So we've got first question from the audience is, you know, a lot of our facilities have long stainless steel lines. So if you're unable to disconnect those lines, which would obviously make scrubbing difficult, what's the best or recommended way to prevent or completely remove the biofilm in this case?
Well, in this case, now, if the pipes are long, if it's a one pipe long that has no rubber gaskets in between those pipes, right? It's okay if you can use an effective a cleaning procedure, which you have to have the speed of the solution when you clean with it, a detergent. You have to have a 5 seconds, 5 feet per second, the velocity of the speed of the solution. When you use caustic detergent, it has to be at least 5 feet per second. So, but if you have this, if this long pipe is all connected pieces, you've got to break it down, because those rubber gaskets will have a lot of slime on the inside of them. When you break it down, you'll see a lot of slime, and the slime is a biofilm, and you have to replace them and scrub the gaskets if they're not cracked. But if they're cracked, you need to replace them with the new ones.
Got it. And Adel, while I'm asking you this question, if you want to put your email address in the chat box so that everyone can follow up with you after, because we are getting a lot of great questions.
Okay.
But as you're doing that, I'll ask you this. You talked about the smart technology and ATP technology. So if you could highlight what are the main differences between the two? Is one more accurate or more sensitive to organic matter?
Yes. Okay. So as I said, the ATP has limitations. ATP, when you have a piece of equipment that you detected on it, the biofilm, you bring the swab of the ATP. First, you have to have an instrument. You have to pay thousands of dollars for the instruments and the swabs to do the test. And then when you do the test, you do it on only a small part of that entire surface, 2 by 2. You swab with it and then you use it. So you might have picked a good area where there is no bacteria, there is no food in there, and you left the other areas that are not. When you use the colorimetric technology, the smart technology, you can spray it on the entire equipment and see where the color is changed, and you would know if there is any organic matters or biofilm or not by looking at that piece of equipment. Okay, so it's more effect, and the sensitivity of the product, of the colorimetric product, is higher than the ATP. The color changes with the minimum amount of organic matter on that surface. ATP will require more organic matter to be read through the instrument.
We have a question around, can you discuss, and I'm paraphrasing a little bit, but can you discuss the new technology represented by both back to scan, back to scope using UV light for biofilm detection?
Okay, would you repeat again? But your voice was going in and out.
Yeah, sure. Can you discuss the new technology behind back to scan and back to scope using UV light for biofilm detection?
Honestly, I have not heard of that technology myself.
Interesting.
But I would like to know about it.
Very good, good. Then we've got homework to do, you and I, right?
Yeah, I guess.
We have a question here. Is there a fogging application for your products?
The fogging application, I don't think we have that fogging application for it. But I would not. We need for fogging. I think it's going to be difficult for using this product, because when you fog, you use a very minimal amount of a chemical that hits the surface to keep it kind of still dry, but it's at the same time wet. And this little amount of fog on a biofilm or an organic matter will not be removed, will not help removing. It will require more chemical to remove it. So I don't think we have an approval for fogging application, but I wouldn't encourage it unless it's approved and it's effective. Then we can add it to the label of the product.
Got it. I think we'll take the remainder of the questions offline. What we'll do is when we post this to our IDFA Knowledge Center, we'll have this recording, the slides, and we will also include Adel's contact information so that folks can follow up after this webinar. But I want to thank you, Adel. I want to thank AFCO and ZEP for their partnership, their membership in IDFA. And I want to thank all of our participants today for spending some time with us. Thank you for joining us. Adel's contact information is right there on the final slide. So look forward to joining us for future virtual programming. And thank you all for joining us today.
And please
Adel,
everybody, feel free to contact me if you have any questions. I would be more than happy to help you. And thank you, Tom. And I want to thank again the organization for this opportunity. You guys have a wonderful week.
Thank you. You as well. Bye, all.
All right. Bye-bye.